Hey Scronce, I just woke up from a nap, but this is my video for bio. So yeah. Okay, so my first one pager I made was like 4.1, so like that's what I'm doing first. So carbohydrates are made of CH and O. Their monomer is monosaccharide, and that's like glucose, fructose, and like galactose. I don't know how to say that, but that's what I wrote down. And then it's found in plants as a starch and found in animals as a glycogen. So yeah. And then proteins are made of CHO and S. Their monomer is amino acid, which is like the links. And then I drew a picture of that. That's a protein. And the nucleic acids are composed of CHO, N, and P. And their monomer is a nucleotide. And they have phosphate, dentose sugar, and nitrogenous base. And their bond is a phosphodiester linkage. That's what it looks like. And then a lipid is composed of CHO and P. Their monomer isn't anything. They're saturated, like a saturated fatty acid is all single bonds, and unsaturated is just at least one double bond. And then all lipids are not. The sequence for like the relationship among DNA, proteins, and amino acids is the DNA consists of gene codes for a specific amino acid that combinedly forms a protein involved in trait expression. So that's what that one is. And then how enzymes act as a catalyst for biological things. So biological reactions. Enzymes act by reacting to the activation energy, thereby like increasing the rate of reaction. So that's it for my bio 4.1. This is what my paper looks like. Okay, now we're on bio 101 and 1.2. I'm doing them combined. So bio 1.1.1. I'm just gonna explain all these cells. So yeah, this might take a while. So the nucleus is a double layer covering called a nucleus. It has that, and it's called a nuclear membrane, which has pores to allow the transfer of materials. It's the controlling center of the cell, so like it basically just like is there. It's like it's your brain basically, and it helps transfer genetic information from one generation to the next. Then we have the plasma membrane. It's the membrane found in almost every cell, and it separates like the exterior from the interior. And it keeps toxic substance out of the cell and also mediates the transport of solutes, ions, and molecules. The mitochondria, this is like the real brain. Like the nucleus is just like your side brain. It's the powerhouse of the cell, and it's found in the cytoplasm. It's a double membrane structure that consists of cristae and effluent particles. Next is vacuoles. They are storage, like they store everything, and they're found in only plant cells. They're surrounded by like the membrane of the plant cell, and it's called tonoplast, and they provide rigidity and help maintain the osmotic pressure of the cell. Ribosomes, they're like circle, and they're glands like shaped in the cell organelle that is in the cytoplasm and remain attached to the ER, and they play a important role in protein synthesis. And chloroplast is only found in plants and green algae, and they consist of green-colored pigment called the chlorophyll, and they're involved in the process of photosynthesis. And they help make carbon dioxide into glucose in sunlight. So, yeah. I'll be right back with 1.12. Okay, so some differences between prokaryotic and eukaryotic cells are the cell size of cells. And the eukaryotic cells are bigger in comparison to other ones. Eukaryotic cells have a more defined nucleus, which is surrounded by the nuclear membrane. The nucleus contains the genetic material DNA, and then the prokaryotic cells lack like a true nucleus. Like they don't have like an actual nucleus. Their DNA is found in something called the nucleoid. The cell wall, both eukaryotic and prokaryotic can have a cell wall, but the like what it's made out of is different. Like a eukaryotic cell walls, when they're present, are like made of more like cellulose and chitin, and depending on the organism, prokaryotic cell walls, however, are like made of like peptidoglycan. I can't pronounce that. In reproduction, eukaryotic cells reproduce through mitosis or meiosis. I think that's how you say it. A process involving replication and division of the nucleus. And prokaryotic cells reproduce via binary fission, a process like that. It duplicates the DNA and then divides it into two like identical daughter cells. There's some differences, but there's like different ones. Like some prokaryotes may have specialized structures or organelles, nonetheless. Like most of that is like really it. We're on to 1.1.3, and it's just to explain how instructions of DNA lead to cell differentiation and result in cells specialized to perform specific functions. So this is because all cells contain the same DNA. All cells have the same potential to become any type of cell. However, once it changes, it can't be reversed. So it's that. So as they're made up into similar monomeric units termed nucleotides, the two DNA strands are referred to as the polynucleotides, and each nucleotide is made up of phosphate group and a sugar, and one of the four nucleobases cysteine, guanine, adenine, and thymine that contain nitrogen. So that's really it for that one. So we're on to Bio 1.2, and I'm starting with Bio 1.2.1. So that's explaining how homeostasis is maintained in the cell and within the organism in various environments. So the plasma membrane determines what enters and exits the cell, regulating homeostasis in the cell. An example is in humans, homeostasis in the blood is regulated through a lot of things, kidneys regulate pH and stuff, lungs regulate oxygen, and the temperature is regulated by two things, through skin and your lungs. So basically, with all that being said, the plasma membrane helps maintain solute concentration because it lets it in simple compounds like water, and sometimes complex compounds like sodium, which balances the concentration of what's in the cell and how basic and acidic the cell is. So all that helps maintain homeostasis and things. Okay, we're on to 1.2.2, and that's analyzing how cells grow and reproduce in interphase mitosis and cytokinesis. So interphase is the phase where the cell is replicating its DNA to get ready for mitosis. Then mitosis is four phases: prophase, metaphase, anaphase, and telophase. And then cytokinesis just literally means cell movement. It's after the telophase ends, and then the two cells separate with two identical packets of DNA into individual cells. And that is basically how that happens. Okay, we're on the last thing for Bio 1.2, and it's Bio 1.2.3. And that is explaining how specific cell adaptations help cells survive in particular environments. So let's get to it. Organisms develop adaptations to survive. When you focus on unicellular organisms, which is specified in the packets, they have vacuoles. This helps maintain balance in a unicellular cell. Another structure is that the structures of these are cilia, flagella, and stuff like that. Cilia are hair-like things that help gather food and move materials. The flagella helps it move, and everything that's extra on the cell helps it survive in that new adaptation. So unicellular cells determine its environment and know where it's going, so it knows where it's going to end up, so it adapts to what it's going to be. So yeah, we're halfway done, so that means I'm halfway on my way to bed by now. But I'm on Bio 4.2, and we're starting with Bio 4.2.1, and that's analyzing photosynthesis and the cellular respiration in terms of how energy is stored, released, and transferred within these systems. So photosynthesis is the process of converting light energy and transforming it into chemical energy and storing it in the bonds of sugars. Photosynthesis primarily takes place in the leaves of plants, which photosynthesis only happens in plants. So yeah, sounds a little backwards. All photosynthesis occurs in the chloroplast. The energy harvested by the plant is stored by creating ATP. The energy is then released and formed into oxygen, and cellular respiration energy is converted from the chemical bond glucose to a reusable form of ATP. The energy is released when the bond is broken between the two phosphate groups, and that's the entire process of that. We are on the last thing on this paper, and it's Bio 4.2.2, and that is explaining ways that organisms use released energy for maintaining homeostasis, which is active transport. So yeah. It uses ATP to pump molecules against / up the concentration gradient. The transport occurs from a low concentration of a solid to a high concentration of it, and it requires cellular energy. Types of transport is cell membrane, sodium, potassium pump, and yeah. The functions is the main purpose of it is to transport molecules through the cell membrane against the concentration gradient. So more of the substance is inside the cell or outside the cell than normal, disrupts equilibrium established by the diffusion. And types of stuff they transport is proteins, ions, large cells, and complex sugars. An example of it would be sodium, potassium pumps, and secretion of a substance into the bloodstream. It's important in eukaryotic cells. as like amino acids, sugars, and lipids need to enter the cell by protein pumps, which requires active transport. These either cannot diffuse or diffuse too slowly to be there in time before they die. So this all happens to help move things faster. So yeah, we're done. This whole paper. I hope you enjoyed my video. I'm sorry for anything loud in the background. My house is really loud.